First of all, I would like to thank the members of the committee for giving us for one more time the opportunity to present in front of this nice audience a rather heretic approach that tries to explore and exploit the computational role of non-neuronal cells. So let me give you the main message of this talk right up front. And I want to emphasize that the brain has been computing long before we started recording the electrical activity of its neurons. So this century of neuronal recordings has created the dogma that brain computation equals neuronal processing. However, the last 10, 15 years, due to new imaging modalities, we're able now to see that there are other non-neuronal cells that are at least implicated in processing and learning information in the brain. So the two wings at the Computational Brain Lab are, on the one side, the understanding of the non-neuronal mechanisms in brain function and perhaps dysfunction. And on the other hand, the other wing is to translate this new to us understanding into neuromorphic, or I should say brain-morphic computing. So this talk will evolve around this computational result that hopefully would be fully digested by the time I finish this talk. We got interested in the disruptive potential of a single cell in the brain to detect whether a neuronal network is at the subcritical, chaotic, or critical regime for two main reasons. In fact, both neuroscience and neuromorphic computing are actually interested into this. So there are many experimental and theoretical studies that suggest that the brain actually operates at this critical regime, right at the edge between order and chaos.
The third paper actually says that it's subcritical.
Well, it's slightly subcritical, so it's important for us to actually be able to recognize the criticality. And even for liquid state machines, you can actually understand that if you have a network that encodes the input into its dynamics, if you have a chaotic system that the one state is completely unrelated to the other one, you cannot really do much in terms of encoding the input. So in terms of the applications, we built robotic controllers that are based on spiking neural nets. This is one project that has been greatly facilitated by Intel through the NRC grant. And quite recently, we proposed an SNN, which is essentially a network of 5 subnetworks that make this robot be able to recognize, to learn the environment and recognize where it is, essentially solving the unidimensional SLAM problem. And in fact, this method has been of similar accuracy compared to the state-of-the-art G-mapping method, and has been, when it's run on Loihi, it's giving us considerably less power in terms of, so we have similar accuracy and much less power. We realized, though, that this took a PhD student of mine a good 2 years to build that. So even if you account for the training time, this is a lot of time. So if we keep asking for our robots to do something more, we shouldn't rely much on actually handcrafting SNN and imitating the structure. As I said in the beginning, this neuronal paradigm of studying the brain has been the prevailing hypothesis, working hypothesis for 100 years. And there are good reasons for that. Actually, there are 2 good reasons. The first one is we became really good in recording the neuronal activity. And over the last 15, 20 years, more like approaching this as an evidence-based technique, we also understood that by manipulating the electrical activity of these neurons, you can actually change the overall behavior, the macroscopic behavior of the network. And these are 2 FDA-approved techniques to change the electrical activity of neurons and from that alleviate the symptoms of diseases such as Parkinson's disease with DBS and major depression disorder. Now, if you ask someone how this works, nobody knows. So it comes as a surprise to many to learn that up to 90% of the brain cells are not neurons. These other cells are glia cells and have been remaining electrically silent as we have been recording neurons. Therefore, they're completely absent from brain hypothesis. Now, don't get me wrong. It's not that we did not know about the existence of these cells. This is a drawing by Ramon y Cajal back in 1897 of an astrocyte. About 40% of our brain cells are astrocytes. What we could not see was their function, and have superimposed a calcium imaging video of an astrocyte that was recorded in 2015. And you see that this astrocyte is actually responding on the neuronal activity by changing the concentration of the calcium inside its soma. So astrocytes are not just neurons hanging around, are not just cells hanging around neurons. They're actually interacting with neurons. And in fact, they do three things. The first thing they do, which is going to be important for this talk, is that they listen to the neuronal activity. In other words, they have the same receptors that a postsynaptic neuron has. And whatever transmitter gets diffused into the synaptic cleft has more or less the same probability of being captured by a receptor in an astrocytic end foot that ensheaths the synapse, the so-called tripartite synapse. The other two things that astrocytes do can be captured to a single word. They modulate neuronal activity, and they do so by inhibiting the presynaptic neuron and by injecting their own current, by injecting their own information into the postsynaptic neuron. Interestingly enough, they do so in a completely different time scale. So the time is important here because we built compartmental models of astrocytes. From a neuroscience perspective, what these models do is they preserve the identified compartments inside the cell, as well as the organelles and their dynamics. From a technical point of view, these models are adding an orthogonal dimension in the synapse, essentially introducing a series of learnable nonlinearities. So what we do is we build models that look at how microscopic phenomena can actually change the network behavior. And these are just 5 examples that gave us enough traction to get results and therefore publication over the last 2.5 years. So very briefly, I'm just going to go through each one of them. The first thing we did was we built a spiking neural network, and we superimpose on top of it an astrocytic component, and we let them interact. And the typical way to present this slide is that we put that spiking neural astrocytic network to sleep, and oscillations that have been observed in sleep emerged as a network property. Now, a few months after we archived the paper, there was an experimental group in Hungary that actually devoted a paragraph in their discussion saying that their experimental data actually align with our computational hypothesis. So we started scratching our heads saying, well, if by not doing anything, this spiking neural astrocytic network threatens to answer a question, 100-year-old question, like how do oscillations emerge? The oscillations that I showed you here are preserved even if you cut the thalamocortical connections. Therefore, people have been observing them, but they don't really know what is the mechanistic origin. What would happen if we introduce function to a neural network? So the first thing we did to introduce function is we created a Hopfield net, and we created memories on it and had a sequence of memories. So what the astrocyte did, and I'm not going to go through the details, we presented that last year, is that the astrocyte learned that sequence, and by the time we showed to the network and the astrocyte that was listening to that network the very first memory, by also following the behavioral time scale, it was able to steer the network from memory 1 to the learned memory 2, memory 3, memory 4, and so on and so forth. The other 2 results that I want to present are quite new. essentially what astrocytes do is that they can encode the synchronization of neurons in an area, and they can actually impose synchronization in non-active neurons. And by doing so, they could, and we have always, I have here our model, and these are data that at least align with our computational results. And so if you can have an astrocyte encoding the synchronization and imposing synchronization, you can end up having heterosynaptic plasticity, and therefore having neurons that were previously inactive now actually being active. So just, again, to summarize what we do is that we look at the astrocytic mechanisms that might explain components in brain science, but they can also be used for brain morphic computing. Take, for example, the very first line. One of the biggest advantages of neuromorphic hardware is that it's asynchronous. However, the brain knows that when it's time to do something, it makes some neural areas entrained into an oscillatory activity. There is a good potential here that an astrocyte is actually going to contribute to that. So this starts the main part of my talk today. And this is a picture of this, well, the receptive fields. Its color that you see here is a single astrocyte. So there is this non-overlap in the receptive fields. And we wondered, what does an astrocyte do? Yes?
What are the black dots?
The black dots are the somata of the neurons. So we wondered, what is an astrocyte doing as it listens to these thousands of synapses? Well, we started addressing this question by declaring defeat. It's experimentally, it's impossible right now to record all the synapses in the area. However, the best model that we could use is the so-called Ising or Ising model that has been shown to have brain-like near-critical dynamics. And more importantly for us, it can transition between subcritical, critical, and chaotic state by just tweaking one variable, which is temperature. Essentially, you introduce energy to the system so that it overrides the local connections between the neurons. Well, the bad news about that is that this Ising model is not biologically plausible. So there are many studies that suggest that synapses create spatial clusters. So we incorporated that into our model, and we were able to show, and you can see now that the spatial clusters are preserved in the neural activity. And we did the typical analysis that people have been doing in terms of the Ising model to convince ourselves that we can actually estimate this critical point, so the edge right between order and chaos. Interestingly enough, the results of this modified Ising model, just like the case of the Ising model, show power law scale without this to be actually a sufficient indication that we're actually talking about a critical system here. However, if you compare the brain data with our model, they're similar. This is a slide that I feel I could not hide the math behind it. So what I'm going to do is I'm going to describe what is happening, and you will see the equation that is actually describing what we have. So the first thing is when we have a spike arriving in the presynaptic terminal, there's a glutamate release that gets bounded into receptors in the astrocyte that releases IP3, which is the first molecule, signaling molecule inside the cytosol of an astrocyte, that gets bounded into the IP3 receptor that releases the first puff of a calcium wave. Now, there are more because that calcium actually activates an enzyme that on itself activates another IP3 wave. And we also have Serca pumps that take calcium out of the cytosol. And in the end, we have that calcium wave. So the output of our well, before we talk about the output, we did two modifications in the original model. The first one is we introduced multiple synapses. And the second thing, which is quite important, is we introduced homeostatic plasticity into the receptors here. And we actually followed experimental results that are mentioned here. And this is the learning curve. Essentially, what this curve says that when we have persistent high firing activity, we have much less receptors. When we have significantly low activity in that particular synapse, we have a huge increase in the receptors. So the astrocyte becomes very sensitive of the existence of even a slightly above zero concentration of the glutamate. The output of our model, so you understand that the input is the synaptic activity, the output is the calcium wave frequency. The take-home message of the whole slide, just for the interest of time, is that we did some housekeeping to ensure that we are OK in terms of the biological constraints. So all synapses fired between 0 and 20 hertz. So this is the main result. And you can see that when the neuronal network is operating in the subcritical regime, the frequency is much lower. Well, we cannot see that. Let me try to rerun it. Yep. So you can see that the frequency here is much less compared to the case that we have a chaotic activity in the neuronal network. In fact, you can see that in this critical regime, we have a fast increase of the frequency that is up to 40% compared to the basal activity. So is this magic? No. What is happening, and you can look at this distribution of the rates as we go from the order to chaos. So in the case of subcritical regime, we have a binomial distribution. And as we go into the critical region, we have a uniform distribution, and then it goes to Gaussian. Essentially, each synapse has more or less the same probability of either being active or non-active. So we have this Gaussian. Now, it's because of this disproportionality in the learning curve. So less active neurons get many more receptors. That makes, when we go into that critical and then chaotic regime, makes the astrocyte to increase its firing rate. In fact, since December 2018, we have keep updating a module that we have on GitHub that allows anyone who wants to explore and exploit astrocytes on Loihi. And this talk today actually gave the very first step of a two-step procedure, which is going to close the loop towards what we could call astrocytic organized criticality. So right now, we're actively looking at how astrocytes could actually use their SIC, their ability to talk to a postsynaptic neuron, to actually not only encode whether that network is in the chaotic or subcritical regime, but actually to impose the whole network into the critical regime. With that, I would like to thank our founders for the two wings, Intel, of course, and the students that are pushing their limits and our limits. I'm just here to present their work. Of course, the experimentalists at Rutgers that are actually supporting us. So we go from a wet lab all the way to robots behaving the way that we would expect them to behave. And these are the papers that we have published since 2017. And I would be happy to answer any of your questions.
Thank you very much. Enthusiastic hand going up over here.
Oh.
This is absolutely wonderful,
Thank
really,
you.
and very new, and lots of insights. I already asked you 10,000 questions. I only have one. How do you think this is involved in the wiring of networks? You know, because it seems that this thing is there, and sits there.
Yeah.
And so, so here's the question, by the way. Is there a mosaic of different sizes, or is it just one size of astrocytes?
Oh, yeah. So since we have 200, yes.
So in the retina, for example, the receptive field of every ganglion cell will change from very small to very large.
That's correct, yeah.
And you have like a 15-layer of mosaic all getting. So do you have the same thing here?
Yes. So we have about 200 identified types of neurons, and we're talking about similar number of astrocytes. So astrocyte, just like a neuron, is an umbrella term. What I could say to answer your question is that we're finite people. We only have 24 hours per day. This is a really exciting question. I wish I had the time and stamina and resources to actually do it. I mean, yeah. So what's the effect in terms of neurogenesis, neuroapoptosis, since actually astrocytes have been implicated to have supportive role to neurons.
What's the scale between the smallest receptive field and the largest of these?
So yes, so we're talking about tens of thousands of synapses. And as many things in neuroscience, there's not a consensus between experimentalists. And I would say that the order of magnitude of neurosomata that are ensithed by a single astrocytic receptive field, and this is on quads, is about 7 to 10. And again, don't get me wrong if that number changes in a year from now.
Okay, one more.
You said at the beginning that astrocytes are electrically silent.
Yes.
And then you said that they are responsible for this calcium wave.
Yes.
How do you reconcile that?
So the calcium is inside their body. So they change the concentration of the calcium ions inside their body. But if we stick an electrode next to them, we will see that the difference of the potential between that electrode and the reference electrode is going to be very close to zero.
So the wave is not one of the propagating waves?
Yes, so it's inside their cell.
Okay, Konstantinos, thank you very much. Let's thank him again.